Welcome to our Lancaster Environment Centre online seminar programme. For practical reasons, this seminar session's been pre-recorded and uploaded directly onto YouTube. That means we don't have a live audience to ask questions, but I've asked our speaker to leave their contact information on screen and contact them directly for more information on their presentation today. So without further ado, I therefore hand over to Sarah Lennon of the New Phytologist Trust to introduce our speaker for today and to chair the session.
Hi, thanks, Ali. So, as I said, I'm Sarah Lennon, the Executive Editor at the New Phytologist Trust, which publishes two plant science-focused journals, New Phytologist and Plants People Planet, and which is based at Lancaster Environment Centre. I'm really delighted to introduce Dr. Andrew Groover today, who'll be presenting his talk titled Exploring the Complex Biology of Trees Using Genomics. Andrew is a research geneticist with the United States Forest Service and also adjunct professor at the Department of Plant Biology at the University of California, Davis. Andrew's research explores how molecular, genetic and genomic approaches can help us understand the formation and development the development and evolution of forest trees with a primary focus on wood formation. Andrew has been an editor of New Phytologist since 2012 and head of the Physiology and Development section of the journal since 2016. And in 2018, he became a strategic advisor to the open access journal Plants People Planet. So it's a great pleasure to introduce Andrew, and I'll pass over to him to begin his talk.
Hey, terrific. Thank you so much, Sarah. And let me just get my slides going. Okay, so it's a pleasure to be able to share with you today some recent results from my group. And one of the major challenges that we face is how to understand the very complex biology of forest trees as it relates to subjects ranging from their utilization for forest industry to how forests will respond to climate change. And I want to start by just stressing the importance of genetic variation for forest biology. So genetic variation is, of course, the currency of speciation. And I'm showing here leaves from some of my favorite forest tree species. And as you compare the variation among these different leaves from species ranging from aspen to sweet gum, really these differences in shape are ultimately a reflection of the differences in the genes and the genomes of these different species. But importantly, genetic variation within species and among populations is really the basis of adaptive evolution. So this sort of variation is going to be what determines whether or not a species or a population can adapt to new climate conditions, for example. Now, most forest tree species have abundant natural variation. Most species are highly outcrossing, highly heterozygous, and have large amounts of natural genetic variation. So one of our challenges for forest biology is how to understand this genetic variation, how to measure it, how to ultimately understand and manipulate it so that we can do a better job of managing our forest and conserving forest in the light of climate change. In that regard, quantitative traits are really central to forest tree biology. So as shown in the top part of the slide, most traits for commercial forestry are quantitative traits. So things like yield, wood quality, pulping characteristics, and so on are not discrete quantities, but rather they show continuous variation. And the little histogram there is showing an example of this for a pedigree of loblolly pine trees, where on the y-axis we see the frequency of individuals within the pedigree, and on the x-axis we have wood-specific gravity, an important trait for industry, ranging from low to high. And what you can see is there aren't discrete categories of, say, small, medium, and large, but rather there's continuous variation for this trait within the pedigree. Now, the typical interpretation of this sort of variation is that it's reflecting that the variation is controlled by large numbers of genes, each of which vary within the population or pedigree, and each gene has a small incremental effect on the trait. Now, likewise, most adaptive and ecologically important traits are also quantitative for trees. So, for example, in the picture, I'm showing you some aspen trees in the Sierra Nevada near my home in California that are changing color during the fall. So this sort of phenology trait is quantitative. Also, traits like leaf size, leaf shape, drought resistance, and so on are all quantitative. So in the past, this sort of quantitative complex variation has been very difficult to really understand, certainly at a single gene or even at a mechanistic level. But one thing that's very exciting now is that we have new tools that allow us to begin to explore and understand these complex traits directly in forest trees. And much of this advance has been based on new technologies, notably genomics and other omics technologies, certainly advances in DNA sequencing, and also computational tools. And it's hard to overstress the importance shown here in this cover of Science, celebrating the sequencing of the first forestry genome for poplar, which was published in 2006. This was a real game changer that allowed us now to move from simply kind of describing and breeding experiments, the type of variation we see, as I showed in that histogram, to now being able to really dissect complex traits down to even individual gene levels. So what I want to share with you today is an example of how genomics can be used to dissect complex traits directly in trees, in this case in poplar, is focused on a special type of genetic variation called gene dosage variation. So in this project, I'm going to try to answer broadly two questions. First, how does gene dosage affect leaf morphological variation in poplar? And secondly, how does dosage affect gene expression in both cis and trans? This is part of a long-term project. It's a collaboration with Luca Kamai and Isabel Henry at the UC Davis Genome Center. And Eloise Bastians was a very talented postdoc in the group, and she was responsible for leading the collection of all of the phenotypic data that I'll be showing. She also performed all of the quantitative genetic and bioinformatic analyses that I'll be showing. So first, I wanted to explain what I mean by gene dosage variation. So structural variation and gene copy number variation change relative gene dosage. And I'm trying to show you how that works with a little cartoon in the upper right. So what I'm showing you are, in cartoon form, chromosomes from a hybrid cross between a Populus nigra and a Populus deltoides. And in this case, each little colored circle represents a gene. And in the case of the green colored gene, we have two alleles, and they encode a positive regulator of other genes, so a transcription factor, for example. And I'm showing that as positively regulating the expression of the first gene in that kind of purple color. Now, below the genes, I'm showing a output representing, say, transcriptional output, transcript level, for example. Now, if we look at what happens if we change gene dosage, so if you look at the same cartoon on the right, now we've increased the dosage of that green gene, say, for example, by an addition or an insertion. And now we have 3 copies within that diploid background. So that has resulted in an increase in the expression level of the green gene, but importantly, it's also affecting the expression level of the gene that it regulates from 2 to 6. So obviously this is a cartoon example, And in nature, this is much more complicated. For example, most transcription factors wouldn't regulate one other gene. They would more likely regulate dozens or even hundreds of other genes. So this sort of gene dosage can actually have very complex effects on gene expression and ultimately on phenotypes. Now, this type of variation has not typically been surveyed in plants, mostly for technical reasons. But with advances in recent sequencing technology, it's now recognized that it's in fact very common. So for example, in poplar, a recent sequencing study of multiple species and genotypes identified over 7,800 deletions and 10,000 insertions within this relatively modest sample. And that affected 3,230 genes within the poplar genome, which contains roughly 40 to 45,000 genes. So this is a very common source of variation in nature, but it really is not very well understood. And so one thing that we wanted to do in the project I'm going to tell you about today is try to understand how gene dosage variation can affect gene expression and quantitative traits at a genome-wide level. So for this study, we created a new pedigree, and what we did was we crossed a female Populus deltoides with a Populus nigra individual, and we took the pollen before doing the cross and irradiated it with gamma irradiation to cause chromosomal breaks. Now, importantly for this study, genotypes can be clonally propagated, and this study, which we did in poplar, in fact, would be very difficult, if not impossible, to do in other model plants like Arabidopsis or maize. So we made a large population of these irradiated hybrid poplars, and importantly, each individual is going to be carrying an insertion or a deletion somewhere in the genome. And the graphs on the right are showing you for one F1 individual how we identified where the insertions and deletions were within the genome for each individual. So the middle graph is showing you relative read coverage from genome sequencing of this individual. And along the x-axis are the 19 poplar chromosomes. And what I hope you can see is that for chromosome 1, there's a region that has increased numbers of reads, and in chromosome 3, there's a region that has a decreased number of reads. The graph in the top is showing you that we can also score these reads for single nucleotide polymorphisms from both parents, and we can combine those data to make an inferred karyotype as shown in the bottom graph, where now we can infer that, in fact, in this individual, there's an additional copy of this portion of chromosome 1, and there's a deletion of this portion of chromosome 3. Now, the little cartoon in the bottom is showing you one way that we can utilize these individuals. And here we're pretending that we're looking at a trait tree size. And what the cartoon is showing you is that if we delete this region of this chromosome, we get smaller trees. If we make an additional copy, in other words, we raise gene dosage in that region, we get a bigger tree. And the little graph is just summarizing a statistical approach where on the x-axis we're looking at relative gene dosage, and on the y-axis we're looking at trait values for the same individuals. And here you can see there's a clear correspondence between gene dosage at this region and tree size. So this collection together creates a large amount of variation directly within a single pedigree. Importantly, it's not transgenic and mimics the same sort of variation that can be seen in nature. Now, this is a very complicated, busy graph, but it's trying to summarize all of the insertions and deletions that are found within this pedigree. The blue bars are showing you where insertions are. Green are showing you where deletions are. And this is actually summarizing for all the individuals, nearly 600 individuals within the pedigree. And what you can see is that we've saturated the genome within this pedigree about 10-fold with insertions and deletions. Surprisingly, we're able to delete almost any region of the poplar genome, and it's not lethal. In fact, many of these individuals carrying large deletions, you would not be able to pick them out easily from their wild-type counterparts. So the poplar genome underwent a relatively recent duplication event, and we believe that might be in part why poplar seems especially tolerant to these sorts of insertions and deletions. Importantly, though, the variation caused by these indels does end up being reflected in quantitative traits. So when you measure these trees carefully for different traits, you will see the effects of these insertions and deletions. So these trees were all entered into a field trial. And I'm showing you on the right here some pictures from this process, starting with propagation. So we're able to clonally replicate each genotype in the greenhouse, then move them out to the field, grow them up. And on the left, I'm showing you some of the many types of traits that we've measured, including some of the leaf development traits that I'll show you today, but also biomass production, tree architecture, wood characters, physiology, and so forth. So the trait I'm going to focus on today is leaf shape variation. And leaf shape we chose for two reasons. One is the size and shape of poplar leaves is correlated with both biomass traits as well as ecological traits. So on the right, I'm showing you a very large leaf from a commercial poplar hybrid. And what breeders find is that in general, leaf size for poplar correlates with the ultimate yield and biomass produced. Now on the left, I'm showing you an ecological example of leaf shape variation in poplar. And this is one of my favorite poplars. Populus euphratica is a desert poplar. It grows in deserts in Eastern Asia. And this is in stark contrast to most poplar species, which are water-loving. One thing that's very interesting about this species is shown by the leaves on the right. So during more permissive conditions, it produces these kind of lanceolate-shaped leaves. But during more harsh, dry, hot conditions, it produces leaves that are much flattened with this kind of crenulate margin. So this sort of heteroblasty is also rather unusual, but nonetheless, it does show that leaf shape can play important roles for how trees adapt and respond to the environment. Importantly for this study, leaf shape and size, for that matter, are nice traits for being able to integrate phenotypic and gene expression data, because it's much easier to interpret when and where we should collect gene expression data for leaf shape as opposed to, for example, a phenology trait. So in this slide in the top here, I'm showing you something that we saw right away as we planted these trees out in the field. So on the left is an example of a wild-type leaf from one of the trees in the pedigree that does not contain an indel. Whereas on the right, we have leaf shapes from various genotypes in this population that obviously vary quite dramatically from the wild-type leaf. And this variation is caused by indels. So for this study, Eloise took both previous genomic and phenotypic data, but she also took leaf images of over 600 genotypes in triplicate over 2 years from the field. And on the right is just a small sampling of the actual outlines of the leaves from her photographs. And you can see there's quite a lot of variation within this pedigree. She also produced RNA-seq libraries for 165 selected genotypes. And ultimately in this project, the goal was to integrate both the gene expression data and the phenotypic data to begin to understand how insertions and deletions, or gene dosage variation, affected gene expression and, in turn, the phenotypes. So the traits that she analyzed are shown here. And there were 3 classes. So first, she had a number of leaf shape descriptors, which were agnostic of leaf size. She also had a number of measures that actually looked at leaf size, so including area, width, height, and various ratios of those. And she also measured stomata distribution, so density, nearest neighbor distance, clustering, and so forth. So one thing that was very interesting about this population is we were first looking at these trees in the field. Some of them reminded us of leaf shapes from other species. So poplars within the genus Populus are in the family Salicaceae that also contains salix, or the willows. And so in this first graph that I'm showing you here, this is showing leaf shapes clustered from our population, but also Eloise included leaf shapes from all the other species shown with the black outlines around the periphery of the graph. So most of the trees from our population clustered along with their parental shapes from Populus deltoides and Populus nigra. But for example, if you look at this purple cluster to the right, we also had a large number of trees that shifted towards neighboring species within Populus, and also willows that have these more lanceolate shaped leaves. So this was a very interesting result, I thought, because what it's showing is that by simply changing gene dosage at various positions within the genome, we're actually able to recapitulate aspects of related species leaf shape. Now, Eloise also used the same data to do genomic scans, looking for what regions of the genome had correlations between gene dosage and the phenotype that she was measuring. So in this graph to the left, you can see again a reminder here that what she's looking at is statistical correlations between gene dosage at each position within the genome and phenotypic levels. And the little Manhattan graph just beside that is showing you the 19 chromosomes of poplar and the data points wherever they're crossing that statistical threshold indicated by the blue line indicate regions of the genome where, when we vary dosage, it's affecting the trait, which in this case is a leaf-shaped trait. Now, all the little graphs surrounding there are showing you for each one of these significant genomic regions what happens when you either raise or lower gene dosage in terms of leaf shape. So, for example, in the top left example for chromosome 17, a deletion tends to make the leaves a little less fat towards the base, whereas insertions fattens out the base and makes the tip a little more pointy. I should say, though, these are actually abstractions of shape and not the literal interpretation of the shape. So what this shows then is that for each of these traits, we're able to scan the entire genome to identify specific genomic regions where quantitative trait loci, where varying dosage will change the phenotype. And we can also use those same data to quantify and describe the change of shape attributable to each one of these regions. Now, this is a very complicated graph, that is summarizing a lot of data. So along the bottom, along the x-axis, is showing you positions in terms of the 19 poplar chromosomes. And if you look first at the top part of the graph, what that is showing you are the positions in the genome for dosage-sensitive regions that affect traits. And the traits being shown are shown in colors to the right. So in the kind of orange color, we are looking at tree biomass traits. In purple, leaf stomata traits. In green, leaf size shapes. And in blue are leaf shape traits. So a few things that you can see from this. One is that there are some regions which are highly pleiotropic that affect multiple traits when dosage is varied. But we can also find regions where varying dosage affects only individual traits. Now, a bit more complicated, the bottom part of this graph is actually showing you relationships between gene dosage and gene expression. If you look along the diagonal of that plot, what that is showing you is statistical significance between gene dosage at a given position and gene expression. So for example, if you look down at the very bottom left-hand corner, you can see a dark color there. And in this heat map, what that is showing or telling you is that there's a very strong correlation between gene dosage and gene expression of genes within that region. Now, if you look vertically above that, you can see that there's also a lot of color in the heat map for other regions of the genome. And what this is showing is that for this region, one or more genes within that portion of chromosome 1 is actually encoding trans-acting factors, for example, a transcription factor, which is affecting the expression of genes elsewhere in the genome. And in fact, if you look along the graph, you can see that there appears to be a number of different regions of the genome that are affecting large number of genes in trans. The two little graphs in between, the phenotypic graph above and the gene expression graph below, are showing you coverage. So the blue bars are showing you basically how good of a statistical sample that we had for a given region. And the little peaks just above that are showing you statistical significance for regions for which we believe these are trans expression QTL hotspots. There's a number of different analyses that we're doing with these data, and the steps are summarized here. So first, using the gene expression data, we're able to identify regions that are responsive to dosage in terms of local or cis regulation, where modifying dosage is affecting directly the expression of affected genes. But we're also able to understand the expression of genes that are indirectly affected through transregulation. Now, I didn't have time to show you this, but we've also been using these data to model transcriptional co-expression networks. And we can actually bring together that gene expression data into functional groups within co-expression modules. We can correspond those back to dosage, but importantly, we can also use that information to correlate genes and modules with phenotypic traits. So at the top, back to the actual phenotypes, ultimately what we're doing with our analyses is understanding the cumulative dosage effects on gene expression and how they influence the various phenotypic traits that we're measuring. So some conclusions from what I've shown you. I hope you can now clearly see that gene dosage is a very important source of genetic variation in poplar. And specifically in this experiment, we showed you that dosage can affect leaf phenotypes, including shape and size. Also, that altering dosage can recapitulate natural variation in shape. And dosage QTL for shape and dosage expression QTL can be identified and correlated. And what we're doing currently is we're exploring the details of how dosage variation affects traits through transcriptional networks, and ultimately how groups of genes and even individual genes affect these complex traits. So again, just to acknowledge some of the folks involved in this work, Eloise was responsible for the phenotypic data collection, analysis, and also for the genomic analyses that I showed you. Matt Zinkgraf was a postdoc in the lab who helped establish this initial resource, along with Courtney Canning, who is our greenhouse manager and botanist. This project was started with the leadership of Luca Kamai at the UC Davis Genome Center, along with Isabel Henry. And I also want to acknowledge funding from the Department of Energy and USDA for this project. And I've listed my email here, and if you have any questions about this or any of the other studies in our lab, I would be glad to try to answer your questions. So thank you.
Thanks, Andrew. That was a really great talk, and it's nice to hear you from all the way over in California, and see some of those lovely images as well, particularly on the last slide of lovely. I presume that was a Californian tree on the last, or Californian vista rather, on the last slide.
It is. It's a tree growing up in the Sierra Nevada.
Yep. Yeah, it's lovely. Okay. So I have a very general question. It's sort of really linked to the point you made about one of the analyses that you could do on the poplar, which wouldn't be possible in a non-model species. And it seems that the genomics approaches you talked about have really helped you to undertake lots more research on the forestry species. But what challenges remain in terms of using forest trees as a subject species, and where does do you find that sometimes some work that you would do that would be easy for someone working on a model species are much more difficult for you to do working on forest trees?
Yeah, that's a great question, and one that really drives a lot of what we do. So luckily, I guess I would address that in two parts. The first is that part of why we study trees is they're ecologically, economically important, but also they have really fascinating biology, much of which is either absent or not well represented by models. So for example, maize does not make wood, it doesn't undergo secondary growth, it's an annual. So a lot of what we study in trees is not well represented by traditional models. Some of the challenges, though, include the fact that trees are long-lived, they're highly heterozygous, they're outbreeding, so they tend to suffer from inbreeding depression. So many of the approaches that would be used, for example, in Arabidopsis, where you might make mutations and then want to self the plants and make homozygous mutants, simply they're just not feasible for trees. So we have to be a little bit more creative, and ultimately I think part of our job is to really focus on what is the unique and really important biology that you can't study in traditional models, and then devise new ways of developing systems like the one that I showed today for being able to make those questions tractable.
That's great, thanks. Any questions from?
I should say I don't know much about genomics. My background is in ecology and looking at things I could see with my naked eye rather than the very small things that make things happen. But I was wondering about the characteristics and traits you were talking about were all sort of focused on physical traits of the tree in itself, leaf shape, biomass, those sorts of things. Is there a relationship could you see an application for this sort of work in like preventing tree disease, be that pest or infection?
Yeah, that's an interesting question. So we are actually using the same collection for other studies. So a couple of examples. One is a graduate student in the lab, Daniela, is trying to understand how poplar trees respond to drought using these same trees. So she's looking both at wood anatomy traits that affect drought physiology as well as physiology traits directly. So we are looking at more ecologically related traits as well. We are also looking at disease resistance using the same population. But I think one important departure for forest trees is that so many of the kind of disease and insect pests, typically invasive, that we have for forest trees, often there's not natural variation for resistance because they are invasive. So the tree species haven't seen them before. So that's a huge challenge as far as how to manage forest and individual species in the light of these invasive threats. We can't really use the sort of approach that I showed today to address those problems.
That's great. Thanks, Andrew. I think, Philip, if there's no more questions, I would encourage everyone to contact Andrew by email and check out his profile that's published a few years ago in New Phytologist if you're keen to learn more about Andrew and his research. And also there's a recent virtual issue on wood biology of New Phytologist, which I would encourage everyone to seek out, and if they're keen to learn more about wood biology more generally. So thanks for your time, Andrew. It was really enjoyable talk, and I hope to see you in Lancaster before too long as well, where you can perhaps meet some people in person as well. So thanks, Andrew.
Terrific. Thank you.
Yes, thanks to Andrew and to Sarah for leading today's session. My final note is just to say that our next session in the series we will be in June, on the 10th of June, and we'll be joined by the president of the European Geosciences Union, who will be talking about his ideas for where geosciences research will go in the light of current pandemics and the state of the research. So hopefully you can join us for that as well. So thank you very much.